Okay, I think we're gonna get started. So this is our last session before the poster session downstairs. We're gonna have a quick set of things. So I'll just say, I think most of the speakers are affiliated with Stanford Data Science, and we've of course been talking a lot about the value of the Scholars Program. We also have a postdoctoral fellows program that closed earlier this year, and we're gonna announce the postdoc fellows soon. But there is an open call for data science scholars that's open now. You can apply here. It's also on our website. Probably will be on the front of our website when the conference is done. This is open for existing Stanford PhD students who are finished with their qualifications. So after the first couple of years, finish your classes, finish your quals, then apply and try to be part of this cohort. And yeah, we accept around 13 to 15 people every year for a 2-year stint. It's a 50% funded effort, so your department funds 50% and we fund 50%. We feel like that's a great way to keep you connected to your department and then also provide a little bit of space for more of your research. So learn more here, look at the details, apply. And there are scholars roaming around. They have this little green tag under their name, so you can feel free to ask them questions about the program, how they applied, successful strategies, etc. So I think that's it. With the next session is really short research talks, a little bit longer than lightning talks. We were playing around with calling them thunder talks, but another way you could think of them is just kind of poster previews. This might be a little bit of a preview of what you'll get when you talk to folks downstairs. And I think without further ado, I'm going to introduce Min, who's going to give our first talk.
All right, thank you for introducing me, Chris. Hi, everyone. My name is Min. I'm a fifth-year PhD student in biomedical data science, and I'm jointly advised by Syrine Young-Levy and Raptip Shirani. And so this is my poster. Hopefully, this serves as a good preview for what you should expect. And the title of my poster is a large-scale vision language data set derived from open scientific literature. And so, let's see, let me go ahead. For this talk, the Thunder Talk, I'm going to split it up into 3 parts. So I'll start off with the motivation of why we decided to build a dataset called BIOMEDICA, and then go into how we built BIOMEDICA. And it's a large-scale dataset, so we had unique challenges in building this dataset and some of the applications that were enabled by building this dataset. Okay, so let's start with the motivation. So to start with the motivation, I have to first talk about vision language models. And so this is a class of models where you're able to provide language instructions. So you can maybe ask a question in language or give it some type of instruction, as well as an image. And it's able to do stuff to the image or have now textual understanding and give it an output, also text and images. So as an example, here asking a question, for instance, is one cat behind another? Give it the image, and the model is able to give back, yes, one cat is behind the other. And for instance, I could also ask it to segment the specific cat, and it's able to do that and provide you the image. And so this is something I generated yesterday. I asked ChatGPT, GPT-4o, which is also multimodal. It's able to take images and text. And so I asked it to generate an image of a cat wearing a hat. And far left, you can kind of see that it did a pretty good job. And then I only tweaked a small thing, just the attribute of the hat, right? Just the color of the hat, I asked it to be blue. And it was able to do that. And then I asked it to generate it into a cartoon style, and it was able to understand the semantics of what it means to be a cartoon and actually generate an image in that style as well. And so clearly these models are really getting good at both visual and language understanding. And so because of that, there's a lot of interest in using these type of models in the biomedical setting as well. And so medicine is a very multimodal setting. There's a lot of clinical images, right? Radiology, pathology, right? We think about microscopy images. There's structured data like EHR. There's a lot of reports, right, like radiology reports and things like that. And so it's very multimodal. And people are interested in being able to use these AI to do things like chatbots for patient, right, interactive note-taking, generating a radiology report, so on and so forth, focusing around clinical decision support. But in order to do this, we really need models that are capable in understanding both the visual aspect and language. Okay. So naturally, we get to ask, what drives the performance of these vision language models? So the answer is scale. And what this means is really simply just more compute So more GPUs, more data, and larger models, and so more learnable parameters. And as we scale through these kind of three axes, we're able to see better performing models. Now, of course, our talk is focused on data, and then we have to ask the question, where can we find such a large-scale data set that has both images and text, because we want vision and language understanding. And it turns out scientific articles are a rich source of such multimodal data. Right? If you look at a scientific literature or an article, you'll get the full textual information. You'll also often see scientific images and diagrams. And they're often accompanied by captions that in detail describe what that image is. And not only that, often you'll find within the text inline text references. So these are things like within a sentence, it'll say, look at figure 4. And we're able to get these type of articles at scale from PubMed. And so PubMed is this free online database of peer-reviewed biomedical literature. And there's around 37 million articles, and it covers and spans about 38 different biomedical and life sciences domain. Now, of that, we're going to specifically focus on the open access subset. So this is about 6 million articles, and you're actually able to get the full text article as well as the corresponding images in that article. And what's nice about PubMed is that every year it grows. Every year, around 1.5 million new articles are added to PubMed. And so if we leverage this data set, we're able to get the latest information and research for life sciences and biomedicine. And just another point about why open source matters. So obviously, transparency, where we care about being able to replicate experiments and also understanding what the models are actually trained on. A lot of these proprietary models, we have no idea what goes into the training data mixture. Also, community-driven innovation. If you have these open source tools, you're able to build upon it, improve upon it. So these are all nice features. But I want to emphasize for biomedicine, a big restriction is PHI. Of course, it's necessary, but to be able to gather data at scale, which is necessary for these modern foundation model training, this makes it quite difficult. But scientific literature is not behind PHI, which is what's nice about using scientific literature from PubMed Open Access. So our goal is to democratize access to open source scientific data across the vast landscape of biomedical research. And so to do that, we built Biomedica. So we actually went ahead and processed 6 million articles. These are like raw tar files that we download from PubMed. And we were able to extract 24 million image caption pairs, 30 million figure references, 6 million full text articles, as well as associated metadata. And we were able to annotate all the images as well. This is a really large-scale data sets for biomedicine. And so let's talk a little bit more about how we built this data set. And because of the scale, there were unique challenges we had to deal with. Okay, so this is kind of the flow diagram of what we did, going from the raw data in PMC-OA as well as NTRES. Please come to my poster if you want to learn more about this. I'm not going to go into details now. But the idea is that we extracted all the necessary metadata, captions, and all this text from the raw data, as well as the images, processed them, and uploaded to a platform called Hugging Face, which I'll talk about in a bit. Okay, so these are some examples of image caption pairs you might find in our data set. And some of the metadata that we also offer. So we include the full text article, licensing information so you can be compliant with copyright, things like, let's see, abstract, title, et cetera, et cetera. So this ends up being like 26 different metadata, and so you can really choose to filter what information you want for these articles. We also efficiently annotated all of these images. So not only do we make the images available, but we want to know what type of images are available. And this becomes important later because you want to be able to filter for things that you might care about, right? Maybe you don't want radiology images, you only want pathology images, for instance. And so we did this using, which I can talk about more detail at my poster, but using these strong image encoders to generate embeddings and clustering these embeddings and then having expert annotators actually look at the clusters and annotate them. And that way, if you have a new image, you just have to project that image down to this embedding space, and you're able to just say, oh, it belongs to this class of images. And after doing that, that's kind of the breakdown of what we found in the images. So there's a lot of compound figures. So these multi-panel figures, when you look at figures, it's like panel A, B, C, D. There's a lot more of those than single panel figures. There's also a lot of plots. And so these are things like bar plots, signal plots, line plots. But despite all that, we also see a lot of images like in microscopy, immunoassays, as well as clinical images that we might care more about in the biomedical context. And so we also offer these more popular biomedical domain subsets, so clinical imaging, pathology, microscopy, surgery, dermatology. And this is nice because you don't actually have to download all 24 million images. Maybe you don't want all the plots or bar plots, but you just want to focus on specific modalities. And this could be used to train, for instance, specialist models. So we talked about open source, but just because it's open source doesn't mean it's necessarily accessible. And so we put a lot of emphasis to making Biomedica accessible. And so part of it really was uploading the Hugging Face, but what this means is that first, all you need is like 4 lines of code to fully load, or to be able to load our data set. So it's very simple to use. And the thing I want to emphasize is this idea of streaming. So what this means is that you don't actually need to download the entirety of the data set to work with it. In fact, when we were working with this data set initially, it was around 30 terabytes worth of data. And so it's open source, but not accessible in that no one really has that kind of storage just lying around. And so by streaming, you actually don't need that kind of storage. Rather, as you're training the model, you're able to load the data. And in fact, for us, when we trained our models, we only needed a space of 50 gigabytes to process all 30 terabytes worth of data. And I really do think because we emphasized and put in so much effort into making this data set accessible, we were able to get already over 5,000 downloads on Hugging Face, people using this data set. Okay, so now let's talk a little bit about the applications. And so this is what was enabled by having a data set like Biomedica. So I'm going to talk about 3 different applications. One is an embedding model, one's a chat model, and then a vector database. Okay, so the first application is a set of model called CLIP. And so this is an embedding model. And the goal of CLIP really is to be able to find or create a representation embedding space that is aligned for both images and text. And what I mean by this is, for instance, if you have images of cats and text describing cats, you want them to be close in this vector space. But images of dogs and text description of dogs, for instance, should be further away from that cluster. And so this model learns this thing called contrastive learning and learns this embedding space and has a lot of downstream applications. And so we trained our own CLIP models, and we did cool stuff to the data, like things like filtering, where we narrowed down to just biomedical images after removing things like plots. We also tried balancing the different classes of images. And we also tried training on everything. And we were actually able to achieve state-of-the-art performance compared to previous models, CLIP models, that leveraged the same data source. Of course, they trained with different pre-training strategies as well as different scale of data. But we were able to, across these 6 biomedical domains representing 40 different benchmarks, we were able to outperform a lot of these models. And the next application is a chat model. And so kind of going back to the motivation where we had a model where you're actually able to interact, provide instruction, get back an answer. We also wanted to do that for our leveraging Biomedica. And so the model we chose is called Small VLM. It's also from Hugging Face. And what's nice about Small VLM, it's fully open source. We know what data went into training it. And it's also really small for a vision language model. It's only 2 billion parameters. And so despite its small size, it's shown in the general domain to show quite strong performance despite the size. And so we fine-tuned on the Biomedica captions that we have in Biomedica. And the way we did this is actually a recipe from a work called LavaMed. But we took the captions and we converted them into this format of question and answering. And the answer is very much contained in the caption, and this is how we generate it. And we fine-tune Small VLM on a bunch of these type of data. And once again, doing so, we evaluate on closed VQA, meaning these are multiple choice questions, kind of like this, given the image, question, and answer choices, and the model is able to answer correctly, right? And provide an answer. And so we compared this to other models of similar sizes as well as slightly larger, and we were able to also show strong performance. So I don't really have time. I'm just going to quickly say we also added a vector database so you can do things like RAG and search quickly for both text and images from Biomedica. And good performance. Yeah, okay. So I just want to say thanks to the team, especially Alejandro, who's also here with us today, and James Burgess, co-first authors. And yeah, thank you.
Hi, everyone. Thanks to the organizers, really, for this excellent conference and for having me here. My name is Ellie Abrams, and I'm a postdoctoral fellow hosted by Professor Xingyao Lai and Professor Dustin Schroeder in the Geophysics Department. And today I'm here to talk to you about the importance of implicit semantic context when classifying rare phenomena in large scientific imagery. And so this is just a toy model of a machine learning pipeline. And for anyone who in this room who is using machine learning for scientific applications, we often use scientific requirements to inform some of the decisions that we make throughout this pipeline. It's easy to see how scientifically driven decision-making might inform our data collection, our validation, our inference and prediction. But I'd also like to posit in this talk that scientific context can be integrated explicitly or implicitly at all stages of the machine learning pipeline. In particular, I'm going to focus on those stages in which we typically make statistically driven decisions, and I'm going to spend most of this talk talking about a data pre-processing method that looks to preserve context. And so Min just spoke to us about streaming data, and when working with extremely large data sets, this is often something that we need to do. So anyone in this room who has used deep learning has likely used a GPU chip to do this, and that's because GPU hardware is extremely powerful for complex computations. But one limitation is that GPU chips have limited onboard memory, and this puts a hard boundary on how much data can be processed by a GPU at a singular time, which means that we often have to stream large data sets through our GPU. This becomes limiting because a single scientific image cube can approach or exceed that size of onboard memory. And so here's an image from a satellite from the Landsat constellation that's actually an image of the Bay Area. I've highlighted where Stanford is on this image with a yellow star. And this constellation, Landsat 8 and 9, images the entire Earth in 5,000 by 5,000 pixel scenes with 10 color bands passing through the entire Earth every 16 days. And this mission has provided images at this resolution and cadence for over 12 years. If we were to take a look at the time series right here, in just 1 year, in just this single 5,000 by 5,000 stamp, a single Landsat image cube is more than 46 gigabytes. For context, the Marlow GPUs are 80 gigabytes. So if we're looking at 2 years of data, we've already exceeded the amount that can be held on a Marlow GPU chip at one time. And so for this reason, it's necessary to divide our overall images into smaller chunks so that we can stream them through our GPUs. So let's see how this might be done. Let's take this image and we'll tile it into 4. Right here, I'm actually going to label the Golden Gate Bridge because we're going to track it throughout this talk. And so we've fundamentally altered our image. What was 1 image is now 4 images. Pixels that were continuous in our overall image are now split, and our algorithms don't have notions of that continuity from tile to tile. And so we have to ask ourselves, how does this tiling have any effect on our ultimate classification? And it turns out that, yes, it does have an effect. So let's turn to a scientific use case. I typically work with data from the Arctic and Antarctica, and here we have an image from Landsat of Ellesmere Island, which is an island at the northern boundary of Canada in the Canadian Arctic. And I have 7 land cover classes overlaid. It's immediately easy by eye to pick out where the snow and ice class is, where the barren rock class is, where the water class is. But we might be interested in some of the underrepresented classes. In particular, I'm going to highlight 2 here, polar grassland and polar moss and lichen, which both make up less than 5% of our overall data. But we might be interested in how those particular classes are changing over time, particularly in this era of increased ice melt. So how did the experts who labeled this image by hand find these 2 classes? Well, as is common in nature, these underrepresented classes occur because of specific physical formation pathways that happen due to the configuration of more represented classes nearby. And so when scientific experts come and take this image, they might actually go through and find that grassland and moss and lichen more easily because of what they expect to see between water, ice, and rock. And so as we're slicing up our images, we want to make sure not to slice away those important contexts. But at the same time, we don't know where these underrepresented classes are, and we want to automate our slicing, because if we were going to go through and slice in such a way that we knew preserved the context, we might as well save time and label the image by hand. And so we're not the first people to ask this question. In a paper famous for something else entirely, for anyone here who has heard of the computer vision method called a UNet, Roneberger et al. Proposes also a tiling method that currently is considered the state-of-the-art tiling. And in order to preserve context, if we turn back to our initial image right here of the Bay Area, Roneberger proposes that you start with your tiling window, and then you slide it over an X by 50%, which now overlaps with those two tiles by 50% that would have made that upper row. Now you slide it down by 50% in Y, and slide it diagonally by 50% in XY. This has the added benefit of preserving multiple contextual windows while also extending our training set. So now we have more data to train on. But there is one catch, and that's that this method actually introduces redundancies into our data set. So if we turn back to the Golden Gate Bridge, which we highlighted before when we tile, you'll notice that before it was represented one time in our data set, and now it's represented 4 times. And additionally, it's represented in the same exact orientation as before. And so this has extended our data set, but it introduces redundancies, and those redundancies fundamentally alter the distribution of our training set. And so we thought, what could we do to preserve these multiple contextual views without creating these redundancies? It turns out there's a neat trick that we can use if we know we're employing computer vision, and that's that most computer vision methods at some point have some convolutional layer somewhere in their architecture. And for convolution, if we permute our input matrix in such a way that it's mathematically distinct from another input, convolution now sees this as a mathematically distinct input and therefore removes redundancies. And so we decided to exploit this and do the same tiling approach, but as we slide our window over so that we have overlapping pixels, every situation in which we end up with overlapping pixels in a tile that has those pixels in another tile, we run a simultaneous, distinct mathematical permutation. So let's take a look how this works again with our tiling. Here I've highlighted the Golden Gate Bridge in a pentagon so we can see how this permutes as we go. We slide our tiling window over by 50%, but simultaneously rotate this tile by 90 degrees. This is mathematically distinct. Now we slide our window down by 50% and simultaneously rotate by 180 degrees, also mathematically distinct. We slide it over diagonally, rotate by 270 degrees. And we find specifically 8 possible permutations that are still physically realistic to observations we'd expect out of our satellite, because we don't want to train on data that the satellite is not likely to observe. And this now extends our data set by nearly 8 times and also provides a completely non-redundant training set at the same time. This concise approach significantly improves the detection specifically of rare classes, so those classes that we were interested in, from semantic context alone. And so we test this approach on a UNet task without tailoring our architecture or optimization to address class imbalance. So we lift the same UNet from that 2015 paper. And what we find is that this preprocessing method alone increases model performance by nearly 16% on these rare classes that make up less than 5% of our overall data. And so if you take a look here at the right column, which is labeled lichen and moss, you'll see that our method, which we've nicknamed flip and slide for anyone who used to play on a slip and slide when you were young, and if you take a look at this method here, you'll see that it does a much better job of approximating the true underlying distribution of that rare class. We wanted to make this method accessible so it could be easily inserted into any machine learning pipeline in Python. And so we published it as an open source package. It's available on the Python package index. You can use it just by running pip install flip and slide. You have these 3 lines of code, and you can output either NumPy arrays, TensorFlow tensors, or PyTorch tensors. So it can insert directly into one of your pipelines. There's a quick start guide here at this QR code. And now, next, I'm just going to give you a quick teaser. This isn't present on my poster, but please come to chat with me about it. This is current work that I'm using Marlowe now to do. So using this flip and slide method to prepare my data in a case of an extremely underrepresented class, I'd also like to posit that when we are searching for very underrepresented things, scientific context can be explicitly integrated at additional stages of the machine learning pipeline in order to improve our detections. And so here's a different Landsat image. Now we are turning to the West Antarctic coastline right here. And I've highlighted a feature that's called a polynya. And a polynya is essentially an area of open water. This type of polynya, sensible heat polynya, opens right at that coastline, and that's due to its formation pathway. It opens a hole in the sea ice, and you can see the open water there. But in this image, I've highlighted 3 polynyas for you. It might be hard to see them in these red squares, and that's because these polynyas make up less than 0.008% of all the pixels in this image. Additionally, to date, of this specific kind of sensible heat polynya, we only have 1 that has been characterized in time for more than just a few years at a time. And that's because this really becomes a small object detection problem. So how do scientific experts, when they created that study, label these satellite images? Well, it turns out that they exploit, again, that semantic context. So this is work thanks to Ojin Lee, a student here at Stanford, who labeled this image. And Ojin found the polynyas by looking specifically for that boundary I mentioned before, that boundary between sea ice and ice shelf. And that's because, if you see here highlighted this basal channel, these kinds of polynyas physically occur only at this boundary. And so we thought, how could we implement an approach that takes advantage of these much more represented classes? Sea ice and ice shelf both make up about 25% of the overall pixels in this image. And so we created a multi-stage method that actually allowed us to label the more represented pixels in a scene, and then optimize for the detection of polynyas based off of their similarities in spectrum and texture to other areas of open water, specifically where we'd expect them to occur. And so in this way, we create a prior of sorts within our vision methods. I'd really like to thank all my collaborators here. I see Josh Ryan's in the audience, actually. Josh is a grad student here at Stanford, as is Ojin Lee. And I work here at Stanford with Professor Xingyao Lai and Dusty Schroeder, and also closely with my collaborators elsewhere, Tasha Snow, Matt Siegfried, and Fernando Perez. This is an extremely interdisciplinary team made up of physicists, statisticians, computer scientists, oceanographers, and glaciologists. Often to do data science, as everyone here knows, you really need that interdisciplinary team to participate. I'll leave up my key takeaways, and thank you so much for listening.
So how are you going to expand this methodology to image segmentation rather than object detection?
This is a great question. Yeah, so we've been thinking about that a lot because segmentation becomes tricky if we try to contextually optimize for when pixels are near each other. If something is large enough, we miss those central pixels, especially something like polynyas, which are small object detection, but some of them are maybe a kilometer in their diameter, some are half a kilometer, some are 3. This is really interesting. And so we've been looking at ways to create different tolerances within it. When we're doing object detection, as we're doing here, we can have a harder boundary condition. But for segmentation, it will be really important to create, rather than that hard condition, a soft condition that allows these notions of context to regularize without fully rejecting instances that don't fit into our boundary conditions.
Hi, my name's Alexandra DiGiacomo. I'm a fourth-year PhD candidate in biology, and I've had the pleasure of doing a year in the Data Science Scholars Program. I'll be giving a poster later today if you want to talk about some of this work, but for the purposes of this talk, I'll give you a broader overview of some of my dissertation research, which was presented at the Data Science and Sustainability Conference recently. So I'll be talking to you about how we're using computer vision techniques to facilitate these rapid insights into marine megafauna ecology. And I'll specifically be focusing on my dissertation system, which is white sharks in Monterey Bay. When we think about ecology, a lot of ecology historically has relied on direct observations of the natural world. So this is the Farallon Islands and a research station here in Northern California. And in the 20th century, researchers were stationed here to directly observe marine mammals, seabirds, and white sharks, which provided a lot of the early historical information that we have on these species. Nowadays, we collect this observational data across remote observing platforms, underwater, aerial, and onboard tags. And this is really transitioning ecology to be a big data discipline. So we've gone from these recorded count measurements to observing across all of these different scales. And in the Block Lab at Stanford, we have collected this data over 20 years across the community, population, and individual level in white sharks. So this leaves us with the question, how do we develop these scalable, non-invasive approaches to understanding sensitive species like white sharks at large body sizes, which make them really difficult to study? And we look towards these efficient solutions in this new era of data-rich ecology. I'll talk about 3 different data science techniques that we use to optimize ecology in white shark studies. The first of which is re-ID, where we try to re-identify individuals by their dorsal fins. In white sharks, we use the animal dorsal fin to identify an individual over time. And this provides really critical information at the population level for us. And how we've done this historically is we print out pictures of the animal's fin that we take from underwater video, and then we lay them all out on a table, we match them, and we provide some mark recapture estimate of how many individuals we have in the population. In a population like white sharks, that number is really important. You can see we have about 200 to 300 individuals, so that number is small. And these species are really heavily managed. So all these different organizations have a hand in managing these populations. Now we're at the point where we have over 20 years worth of data, over 3,000 different images, and so we can't do this manual by-eye matching anymore, and we need these innovative solutions. So we're working with the Lynx Lab at UC Santa Cruz to use vision transformers to take in this visually matched data as training data and develop a model that creates embeddings where we can map these images into coordinate space based on their visual similarity. Then we build this into a recommender system by which, instead of matching each new fin to the entire data set, we match to some top K recommendations. To give you a taste of what this enables us to do, we can start to actually do this long-term monitoring. So in the past few months, we've used this model to match our fins across 20 years. And this is an example where we have individuals on the y-axis of individuals that we've cited at our populations here in Northern California over 10 years. So we can say something about animal behavior, plasticity, and repeatability, but we can also look at an individual site and say something about population dynamics. So this is an example of Año Nuevo, where we can look at the number of new individuals that we haven't seen before versus recited individuals. And we see that in the past few years, we're seeing more new individuals as compared to recites, which indicates that we have some sort of population growth or increased recruitment to the site. So now we've kind of used computer vision to transition us to, instead of every 10 years providing an estimation of the population, we can do this in real time. So we can get this up-to-date information about how our populations are changing without having that lag time of about 10 years. The second project I'll talk about is using semantic segmentation for animal measurements using drone imagery. And in ecology, how we've traditionally estimated animal size is by visual estimates. So 3 trained researchers will look at the animal, provide some guess at how big that animal is, that's averaged and recorded. And that is the only non-lethal way by which we are determining animal age in white sharks, and also their maturation status. So it's a really important measurement for us. And then there are more complex measurements that if we were able to bring the animal on board the boat, but we can't because they're 6 to 20 feet long, we would measure different parts of their girth, which tell us something about animal energetic stores. So we've transitioned now to thinking about how we can use drone imagery to provide these remote measurements of animal body size, whereby we can take a drone image, we can segment the animal without the pectoral fins, we can use skeletonization functions to draw out the medial line, and then we can traverse this medial line and pull out cross-sections that are biologically relevant to animal girth. We can pass this data through photogrammetry equations, whereby we take the altitude and can convert pixels to true measurements, and then take the ratio of these measurements to get a body span ratio, or relative girth. We have now used this model, which is performing very well and predicted on over 2,000 images over the past 3 years. And we can look at our 3 different field sites in and around Monterey Bay and compare the size distributions of our animals. So in my dissertation research, I'm thinking about how we characterize size structure across these sites. And what we're initially seeing is at our warmest and most protected sites, so that site in light blue, which is Santa Cruz, we see our smallest individuals. And then at this red site, which is our coldest and most exposed site at Año Nuevo, we see our largest individuals. And then to give you a sense of how we're using the body condition metric, we can look at how body condition or relative girth scales with total length, and we see this kind of like anomalous point here. And when we look into what that anomalous point represents, this is actually a malnourished individual that was entangled that we observed in 2023 in Santa Cruz. And it gives us a nice proof of concept that we can use something like this body condition ratio to approximate animal health and detect malnutrition. The final data science application that I'll go through today is we're using pose estimation for computing animal kinematics from drone videos. So we use animal locomotion in fish science generally to quantify activity, approximate energetic costs, and identify different behavioral states. And how we do this with small animals is either in captivity, we use flumes or swim tunnels, whereby we pass water at a controlled rate over the animal and we can observe their kinematics. In recent years, we've transitioned to using onboard sensors, so we can use tags with triaxial accelerometers to record this type of information. And this tells us something about how much energy does a white shark spend in a day. And we can start to identify their resting states and also pull out key behavioral states like foraging and disturbance. So now we're using drone videos to track key points along the body. We can use Deep Lab Cut and a ResNet to track these points across video frames. And then we can use the relative position of those frames to inform basic kinematics like tailbeat frequency and amplitude, and even more complex kinematics like regional flexion across the body. And you can think of that kind of like your step length and your frequency of stepping. So these are really important variables for telling us about animal energy expenditure. Then we can take all of these different videos of white sharks that we have, pull out the kinematics, and create distributions across a population. So now we were deploying maybe 5 biologgers on this entire population, and now we can actually apply this video approach to over 100 individuals. So we've mapped over 100 individuals over the past few years, and we can pull out tailbeat frequency, tailbeat amplitude, and this is head amplitude as well. So we can start to develop baselines for animal energy expenditure and locomotion at different body sizes. So just to recap, in leveraging data science for ecology, we have developed these machine learning and computer vision pipelines that really support scalable analysis for us in these really complex ecological systems. The first project demonstrated this minimized data to insight latency, which enables responsive decision-making and up-to-date science. The second project tells us something about how we can make more robust inference about these measurements and population-level patterns of structure. And then the third project really demonstrates the expanded scale of analysis as compared to our previous approaches. With that, I'll take any questions, or you can come visit my poster. Thank you.
just to go. So you're using this approach very interestingly for sharks, and so I guess one of the assumptions is that you can see them because they come up on the surface or not that deep. But there are also a lot of species that kind of hang around more kind of in the depth of like, I don't know, 60 feet, for example. To what extent do you think this method will at some point be generalizable also to other species that are more hidden in the water?
I think remote observing platforms at depth are getting much better, like automated profilers and ROVs that are basically like performing entire plans in Monterey Bay and are capturing white shark movement at depth without us actually piloting a drone at the surface. These approaches, especially the drone-based approaches, have obvious limitations to the surface. I think where we're heading, and part of my dissertation research, is thinking about how we combine both onboard sensing, so attaching biologgers to the animals, and something like observing via like remote underwater video or drone imagery to get a more comprehensive picture. So we can see what the animal's doing at depth, but also when it's readily observable at the surface. Yeah, thank you. Yeah.
Are you aware of any computer vision applications that have been used in beach safety in places like Australia? Because I know that that's always a challenging issue.
Yeah.
And there's a matter of protecting the sharks and things like netting are dangerous to sharks. And so
Yeah.
I'm just curious whether this has been applied in that context. And my second question is, do you ever get in the water with the sharks?
I think some of this basic research is motivated by a few different goals. One of them being like some sort of informed management. I think sometimes there can be applications to beach safety. Typically in this system, that's not a problem that we're encountering very much. In Australia, it certainly is, and there is they have automated drone-based surveying where they do mainly object detection. They want to know when a shark is in the area so they can alert beach safety protocols. We kind of like work farther upstream than that, where we're providing some of the basic information that then can be adapted. And the answer to the second question is no, not typically. Most of this, so that underwater video is done via a camera pole that we submerge in the water. Thank you.
Hello, everyone. We're almost there. I'm a little bit of a fish out of the water here. This is not my field. What I'm showing you before is how to surveys meeting satellites or how to measure experimental outcomes in the field for cheap. I'm a fourth-year PhD student in the Earth System Science program advised by Marshall Burke. I'm usually measuring impacts of climate change, particularly fire ecology. So I'm presenting something today that has absolutely nothing related to it, and maybe it speaks a little bit of the quality of it. This is what happens when you trust too much in your abilities to do things in 2 weeks. But let's go back to what I want to present today. Why do we care to do field measurements for cheap in the field? Well, in wildfires, there's a lot of things that we don't know. But one thing that is like particularly quizzical is like how people behave when there is like the presence of different like natural impacts. This is in Oregon. This is in the middle of one of the wildest wildfires in Oregon, and you see these guys just like, someone there is like looking at the fire. There's these guys playing golf. They should be in their houses wearing a face mask or something, given like the crazy amount of PM that they must inhaling there. But this is one of the reasons why we need to do this. We need to learn how to do fast interventions to inform climate adaptation policy. Currently, we don't have a lot of evidence of what matters or how do people adapt to climate change. We don't have any experimental data, and we have some good observational evidence, but that observational evidence, it's only telling us that we're not adapting. So it's still kind of like an unanswered question from the gold standards of causal evidence. So we need to understand this because we need to adapt. I know this government tells us that it's not such a thing as climate change, but there is climate change, and we need to understand how to survive it. And of course, this is not going to be easy. So rivers of ink have been written on how to estimate wealth or similar remote sense outcomes of wealth from satellites. So these are different papers that actually have come out from Stefanos Ermon's lab, Sustain Lab, during the year. So they have used nightlight data to estimate wealth in Africa. These papers using DHS surveys to have like labels to understand how much assets people have on the ground, picking up from satellite imagery. And more recently, people have tried to do the same thing that we're trying today, which is estimating RCTs using only satellite data. So a possible way to start this is by using embeddings. Minson like shows you how we can create text embeddings from bioarchive papers. What I'm going to show you today is a little bit similar, but we're using images. So this is a recent, well, not that recent, but almost recent paper from Esther Rolfe in Nature Communications called Mosaics. And what they're trying to do is like using a self-supervised way to build featurization from images. So I'm going to focus a little bit on how to build those embeddings and how can we use this in a causal inference setting. So embeddings are very powerful. You have seen it with text. Just 2 presentations ago, we saw that we can have vector databases using these embeddings. They can also do QA. You can train them for a lot of things. The recent architectures have proven to be incredibly good at achieving good representations of different modalities of data. And in vision, this is not the exception. We also have seen how masked autoencoders and other models have also performed very well in few shot abilities. This is meaning basically you don't need a lot of labeled data. If you have a good pre-trained model, you're able to adapt it to something new with just a few data points. This is not something that also I created out of my head. Like there's plenty of companies now taking like a lot of satellite imagery from Sentinel-1, Sentinel-2, and Landsat, and just embedding it and just selling these access to these databases to do different crop type classification or estimation of population. These are just a few companies. There's like many of these doing this. Actually, the bottom 2 are from the European Spatial Agency, like startups. Okay, so the setting here is that we somehow convince Ted Miguel at Berkeley, a development economist, to give us data from GIF directly. So we have access to one of the largest randomized controlled trials in West Africa. This is in Kenya, in the Siaja region, one of the densest rural regions in the world. This experiment started in 2014, and they gave $1,000 to each household. This is 75% of the annual household expenditure. And what they found is that a lot of people will invest this money on roofs, which is a pretty interesting outcome. So this is the outline, this is the timeline of how these interventions and how the money was assigned and how many months it took to collect all the data. You can see that for an intervention that took only 1 month, data collection process is extremely long. And this is exactly the thing that we want to make cheaper and to make faster as well. Okay, so this is how the Siaja region looks like. I don't have a pointer. This is Kenya, and each point there, each black dot is a treatment village. Treatment villages are not how you imagine. So there is no clear boundaries of what a village is in Kenya. So basically how this works is that a enumerator comes, the enumerator will talk to the chief, and the chief will tell him like, well, these are my houses. And then the enumerator will go, and they will probably get lost, and they will like take another house, and they will get confused. So there's a lot of like misclassification of these villages. There's no clear lines of demarcations of them. So this makes our work a little bit hard. I'm gonna show you how we try to solve this. So imagine I can divide the space of Siaja in like a beautiful grid. And in this grid, I'm gonna just classify a grid that is in treatment, a grid that is in control. This is not as easy as it looks like, since I just told you that these villages have not clear demarcation lines. A lot of the time I will have a time in which this grid will have a few houses that are treated, but other ones that are not. That makes things a little bit more gnarly than like in the normal RCT setting. So I will just call these my propensity score and just go with it for a sec. So to build embeddings, there's a lot of like models out there that show us how to do this. One of the issues that we tackle here is that we have images coming from different resolutions. A lot of the pre-trained models that we have are trained seeing images from Sentinel at 10 meter. But for the scale that we want to do measurements, we need to go even further in terms of resolution. So what we're going to do is we're going to take this 1 kilometer square and we're going to embed it. We're going to take the features out of that image, and we're going to try to build these embeddings using, for today, two models. I want to focus on a scale May, which is an agnostic model that allows me to play with the resolution. So even if it has seen 10 meters, I can pass an image of 3 meters, and I'm still able to get meaningful embeddings. And the second one is mosaics, which is like pre-trained. It doesn't need training, actually. It's just like a featurizer. So it's very powerful and has shown like incredible results in other applications. So this is the Siaja country. This is an image from Planet. Planet is a company that currently has a very nice agreement with Stanford, so we're able to get their images. These are 4.8 meter images per pixel. As Ellie was saying before, this is like hella big. Very big. So it's very annoying to train. We have to chop it in different ways to achieve what we want to do. So what we're going to do is we're going to block this. It's going to be 1 kilometer. I'm going to go at the grid level. I'm going to extract only one of these guys. This is actually one of our control ones, and it's very hard to see what's happening there. And I think this is a preview of the results. So to get our y-square, we just need to go from our embedding space to the feature space. So this red area here is like where the embeddings live, and I need to go to the outcomes to run actual regression so I can tell the econometricians like, hey, this is what I found. So to that, I need to build a function that maps those two spaces. And to do that, and once I have that, I can measure my treatment effect in the normal average, like different in means estimator that people on RCTs usually use. Once I have that, I can use the propensity scores that I showed you, just using the proportion of house over the number of selected households, and I can like calculate something meaningful. So these are basically the 2 steps that I need. It's like the 2 elements of my recipe, a good y-hat, and how to estimate the treatments. And everything hinges on how we can do the y-hat. So for now, I'm going to try something quick. It's something easy. I had a nice video here. Imagine these lights are moving. These are night lights. Night lights are coming from one of our military satellites, by the way, BEERS. And it's measuring how much luminosity there is. We have found a very strong correlation in between electricity and income. So it's very natural to assume that if you have like electricity somewhere, you will have like an increase in wealth. You will have more assets. You will own a phone. You will own like appliances and whatnot. So this relationship has survived a lot in like the development economic setting. Can our estimates, can our embeddings do this? Well, they're very good actually at estimating this, but this comes to no surprise. This is a very easy mode. This is a very easy task they have to do. I'm trying this at survey time, and I'm trying this at the last embedding. The reason why I have these two is because at survey time, I have like not like the quality of the images is not the best, whereas in the last year of Planet has come with better satellites. So I expect this to improve. It doesn't seem to be the case, or just a little bit. Okay, but now the harder mode. The harder mode is like, can I see what I see on the ground? Can I actually see what the enumerators see? And that's where things get harder. So this is my survey area. This is just like a piece of that area of Western Kenya that I showed you. And this is Siaja County. So usually the labels that other papers have used are the DHS clusters. DHS is the Demographic and Health Survey, currently run by USAID. That doesn't exist anymore, so we won't have more labels. But at least until a few months ago, we had like several surveys across West Africa that could tell us where were the things that could work. The problem is we don't have a lot. And that's harder to make inference with only a few observations in this setting. So once I do the same experiment I did with NetLights, what I found is that the embeddings are not that good. I'm crazily overfitting in the train and just completely failing on the test. So this is not exactly what we want to. We wish to have like some signal out of this, but it's very hard to get. One of the reasons why is because doing these on-distribution inferences from the embeddings is extremely hard with just a few number of observations. So we only have like 500 surveys, and if you split that into 80%, you just like are losing a big chunk that is helping you. So, so this is why, you know, there's not much I can do here, but I promised I was going to present. But the work keeps going. This is going to pay my years of my PhD, so I need to keep trying to figure this out in the best way that we can. We have the bottleneck coming from surveys. We can fix that, probably. But we can also fix this with getting better images. So we can use Airbus images or other images that are a sub-meter resolution. Maybe we can also improve the embeddings, and this can also show how like what these companies have to offer might improve what we have, or maybe not. We can test them how good they actually are. Or there's a lot of like plenty of papers outside that can help us as well, like super resolution alternatives. Okay, that's it. Thank you so much. That's it. Oh my God, not you.
In the last part, you showed that you had like very limited number of data points, like 500. And I'm wondering what kind of model you used for that?
Oh, absolutely, yeah, sorry, I completely overlooked that. So yeah, basically the way these papers have done this is just like running a lasso. So you have like old embeddings, like 1024, or in the case of mosaics, you can create as many features as you want. And then you just like lasso select and then double lasso on the prediction. That's how we're doing this, yeah, sorry. Thank you. That's a very good question.
Thank you.
Hi, everyone. My name is Syrine Belakaria, and I'm a postdoc in the computer science department. Thank you for being here and for giving me this opportunity to talk. So today I will be presenting my work, which is a joint work with my advisors, Stefano Erman and Barbara Engelhardt, about Sharpe ratio-guided active learning for preference optimization in RLHF. This is like very different from the previous talks. It doesn't have much specific domain information, but it's more like very computer science, LLM oriented, I guess. You can also come see me later in the poster session if you have more questions. And there is much more information about the paper and the other collaborators in the QR code. So to give an overview about the problem, usually in LLM fine-tuning, there are two different types of fine-tuning. We have supervised fine-tuning, where we have like a prompt and an associated really high-quality answer that we consider a label. And there is another type of fine-tuning that is usually referred to as reinforcement learning from human feedback, or RLHF. And in that case, we will have a prompt, but we will have multiple answers. And the label is basically the preference classification between these multiple answers instead. And usually, in most cases, we are dealing with pairs of data with just preference between the two of them. So in some cases, we want to fine-tune our models on scientific data or sensitive data, and we will have a big amount of prompts and associated answers, and we want to label these preferences. But asking a human to label all of these is pretty much unfeasible, especially if we are talking about scientific data, because like we will need an expert in the loop, and that would be very expensive. So the goal of this work basically is, can we actively select the data points on which we want to collect feedback that would accelerate the learning without having to label every point in the data, and that would be much more cost efficient. So here I will provide an overview about RLHF and the basically work that built on RLHF. So in traditional RLHF, we will have these like pairs of data points that we want to get the preference on, and usually like labels would be collected and the reward model would be learned. The reward model basically is a classifier that would, given a prompt and a couple of answers, would be able to tell which answer is better. And then once we learn this reward model, we use it to refine the actual language model that we are fine-tuning. And this is basically done using this equation here. And if you look at the equation, the first part of it is basically trying to make the language model, which is phi theta here, align as much as possible with the reward model. And the second part is basically trying to make sure that the language model doesn't deviate too much from a reference model so that it doesn't fall in like local minima and like so that it also keeps providing diverse answers. After RLHF took a lot of attention, there came another method that basically is a substitution, in some sense, of RLHF called direct preference optimization. And what this method does is basically reducing this process of learning a separate model and using it later to update our language model to a much simpler setting where like the actual language model can learn from the preference data directly without another proxy model and without an RL loop. So this is done basically by finding the expression of phi theta that solves this optimization problem to optimality, and then from that expression, extracting the expression of the reward model. So now we have the reward model just as a function of two existing policies instead of learning it separately. And once we have that, we can inject that in a binary classification loss function, and we are immediately basically making this here what is usually referred to as the loss function of the DPO, which updates the phi model just based on the win and lose responses from the data. So now suppose we have this large amount of prompts and their associated answers, but we cannot label all the preferences of these answers. Can we use ideas from active learning where we can develop an accusation strategy that at each iteration of updating the language model would score the potential prompts and select a subset of these prompts with the highest score according to this accusation strategy, basically? So the main idea that we thought about is that we want to select the inputs that accelerate the learning process and would make the most impact on the model. And one way to think about that is usually like if we are using a loss function, the gradient magnitude that we will get for each data point is kind of an indication of how much update will happen to the model. And we can use it as an indication of the uncertainty of the model about the current data point. However, if we actually want to use the gradient as our indicator, then there are quite few issues that we will have to overcome to be able to do that. So the strategy we thought about is using the gradient magnitude derived from specifically the loss function of the DPO equation. And if we look at that equation here, we will see that, in fact, the gradient magnitude when the first response is preferred to the second is different from if the second response was to be preferred to the first response. And that's kind of a dilemma because we don't know the preference in advance, yet we have two gradient possibilities. So in our accusation strategy, we need to account for both possibilities. And the question was how to combine them. One idea was to compute the expectation, to treat this gradient magnitude as a random variable and compute the expectation over it. And another idea was to use the variance. The expectation specifically is really can be actually a bad metric in multiple instances. So if you think, for example, about a case where one answer, Y1, let's say, is like a sensible answer, a good answer, and the other one is gibberish. And if we compute the case in which like the gibberish is preferred to the good answer, the gradient magnitude can be actually really big because the model will think this doesn't make sense, that then it wants to update that. But in reality, once we give that to the expert, they will not label it in that way, they will label it in the opposite way. And in the opposite way, the gradient magnitude will be really small because obviously gibberish is not going to be adding any value to the model. So one thing we should avoid, in fact, is this case, the case where basically one of the labeling cases would not add much learning to the model, even if the expectation could be really high. And then from there, we thought about this tool from financial statistics that is usually referred to as Sharpe ratio, which evaluates not just the expected value of an investment portfolio, but also the risk. And it is the ratio between the expectation and the variance of different outcomes of an investment. So here we want to maximize the expected magnitude of our gradient update, but also reduce the risk of getting a small gradient update if a certain preference or certain outcome is preferred. But still, even with that finding, this method or this equation basically here will have a big computational bottleneck. And the main bottleneck here is that we want to compute these gradient magnitudes for each data point, basically, that we have in the data. This means that to be able to estimate this accusation function, we will have to backpropagate through our language model twice the size of our data set. So like one when the first case is the first preference is given, and the other when it's flipped. So actually, like backpropagating even a single time on data points individually and with these language models is extremely hard, both in memory and time, because we will have to preserve the whole graph of the neural network in memory for each data point. But then we found out that there is actually a link between the gradient magnitude when we have the first set of preference and the gradient magnitude when we have the second set of preference. And then those two gradient magnitudes are actually linked to each other with just a multiplication factor, gamma here. And gamma doesn't depend on any backpropagation process. It is just an equation that is dependent of the reward model, which is much easier to compute. So now if we look back at the equation, if we inject those relationships in the Sharpe ratio equation, we will see that the gradient magnitude actually reduces from the equation completely. We can remove it. So now we have the ratio that links the expectation and the variance to each other without necessarily having the burden of extracting the gradient magnitude for each data point twice. Here, P1 and P2 are some probability of how much the first answer would be preferred to the second answer and vice versa. Usually we can assume that we don't know that because we actually don't know. That's the whole point of presenting them to an expert. And if we assume that, then P1 and P2 will both be 0.5. And if we set them to those values, we will get even a more simplified equation of our scoring mechanism that is pretty easy and fast to compute. But we can also set a prior on what P1 and P2 could be. And we actually did that in our experiments. We tested both cases. And in that case, we will have to use the full equation. So we ran multiple experiments with several language models, with also several data sets. And we made sure that the language models had different sizes so that we can see the impact when the size of the model grows. We tried to test these methods on very limited number of data points. So if you look at the x-axis, you will see around like 28K data points. This is by the measure of language models usually considered really small number of data points. And if you see the blue curve is the first method, which is like the very simple version of Sharpe ratio where we don't assume any prior on the probability of preference. And the second one, which we called weighted Sharpe ratio, it's the one that we assume some form of prior knowledge about our preference, which we extract from the actual language model, actually. And the red curve is the baseline, where we just sample data at random, basically from the data set. And we have seen Actually, the metric that we are computing here is called win rate over the chosen. So basically, we have generations from these models, and we compare these generations to some ground truth, really high quality answer in the offline data set that we have, and we compute how many times, basically, the model wins. And these are really, really high quality data sets. So even if there is like a small win, it would still be considered good. And we can see that there is some improvement, especially with such limited data. But we can also notice that injecting any kind of prior into the preference doesn't really bring us much advantage in this case. Yeah, so with that, I will mention some of the takeaways. So we were able to find that active learning with direct preference optimization specifically can lead to some sample efficient fine-tuning when the data is expensive. The Sharpe ratio of the gradient magnitude provides actually an effective measure of risk in case of unknown preferences. And leveraging prior information about the preference does not always lead to better active selection of the data, which initially we expected it will provide some enhancement. Thank you.
Okay. Hi, everyone. My name is Sophia, and I'm a fourth-year PhD candidate in the Department of Statistics. Thank you so much for the opportunity, and it's truly my honor to be here. Today, I will be talking about towards faithful synthetic data generation via penalized optimal transport network. So I will begin by introducing the concept of synthetic data. Synthetic data is artificially generated data that mimics real-world data sets without making prior assumptions. So how can we do this? Well, we can use powerful machine learning models to learn the data distribution without imposing too stringent assumptions. And a typical synthetic data pipeline looks something like the follows. So we have nature that outputs real-world data. We gather and we feed them to our favorite machine learning model, which allows us to generate synthetic data. And we can either combine the synthetic data with the real-world data, or we can directly perform downstream analyses using the synthetic data. A popular generative model for generating such data is called the Wasserstein Generative Adversarial Network, which we will abbreviate as Wasserstein-GANs henceforth. The architecture of Wasserstein-GAN look like this. We have a low-dimensional random noise vector, usually sampled from a simple and known distribution such as the Gaussian distribution. We push the noise through a sequence of forward neural network layers that comprise the generator network, and we obtain a fake batch of images or fake batch of data. And to compare how similar our generated data is with the training data, we will make use of a discriminator network that seeks to output higher scores for the real data and lower scores for the fake data. These scores allow us to approximate something called the one-Wasserstein distance, which measures the minimum transport cost needed to move mass from one distribution to the other. And with the approximate with the approximate one-Wasserstein distance, we can then perform two steps of back propagation, one with respect to the generator network and one with respect to the discriminator network. However, a drawback of Wasserstein-GANs is that they're notoriously difficult to train in practice. And this is primarily due to two reasons. The first reason is because we have an adversarial formulation in which the generator network and the discriminator network both engage in a zero-sum game, and this leads to unstable training dynamics. The second is something called the curse of dimensionality, and this happens because the Wasserstein distance is intrinsically unstable in high dimensions. So together, because of these two causes, the Wasserstein-GAN frequently exhibits an undesirable and pathological behavior called mode collapse. So what is mode collapse? Mode collapse happens when our generator network only produces a limited subset of outputs no matter how much variation we inject into the noise. So this means we will only obtain generated samples with reduced diversity. Here's a toy example demonstrating mode collapse. So in our observed data, we have two groups of stick figures. We have the majority group with arms down, and we have a minority group with arms up. In an example synthetic data exhibiting mode claps, we would only observe stick figures with arms down. So that means we have completely lost access to the minority group. As expected, this can lead to serious consequences for downstream analyses and might even undermine the validity of the conclusions drawn from using the synthetic data. Now, mode claps is used quite generally in the literature, and we classify mode claps into two types. Type 1 mode claps refers to mode dropping, and this happens when the generator fails to assign adequate weights to each cluster. So in practice, this means that we will see significant misrepresentations of subgroups in heterogeneous populations. In the plots here, you see that the true data consists of 3 clusters. We have 1 majority cluster and 2 smaller clusters. However, the Wasserstein-Gann generated synthetic data that failed to allocate enough weights to the minority clusters, and hence we don't see the corresponding contours on the right-hand side. Type 2 mode collapse refers to support shrinkage. And this happens when the generator network fails to adequately capture the tail behavior of extreme outliers. And in practice, the tail behavior are of specific interest because they capture important extremal information. In the example here, I have colored the real data in turquoise, and I have colored the synthetic data in orange. And we can see that the support of the synthetic data is restricted to a smaller subset compared to the real data. So if you compare, the real data is from -3 to 3, but the synthetic data only ranges from -2 to 2. The takeaway message here is that if we don't regularize in the right way, then the model will be prone to mode collapse. To this end, we propose something called penalized optimal transport network, which we will abbreviate as POTNet. So recall that Wasserstein-Ganns were difficult to train due to 2 reasons. The first is the adversarial formulation, and the second is the cursor dimensionality. Well, we will tackle these 2 drawbacks one by one. So first, we propose removing the discriminator network and thereby eliminating the 2-component optimization structure. Second, in order to mitigate cursor dimensionality, we can incorporate penalties for mismatch along each axis-aligned univariate marginal distribution. So let me demonstrate with the figure what I mean by these 2 changes. So this is the architecture of the original Wasserstein-Gann. We will first eliminate the discriminator network, and we can do this by using the Monch-Kantorovich primal formulation. Second, we introduce a marginally penalized Wasserstein distance, which we call MPW distance. And this distance leverages the marginal penalty as regularization in order to improve the overall alignment of our joint distributions. We can actually exactly evaluate this MPW distance using an optimal transport solver. And afterwards, we simply perform 1 backpropagation step with respect to the generator network because now there's no discriminator. So this is the POTNet architecture. Theoretically, we establish the generalization bound and convergence rates of the model. Moreover, we're able to show that POTNet provably mitigates type 1 mode dropping, and POTNet mitigates type 2 support shrinkage. Okay, so let me present to you some empirical evidence demonstrating that POTNet does mitigate mode collapse. In the first example, I have a 20-dimensional Gaussian mixture from 3 Gaussian distributions. I'm comparing POTNet against Wasserstein-Gann, and I'm comparing it against this model called OT. So OT shares exactly the same network architecture as POTNet, except for I do not incorporate the marginal penalty. So we can see here that the true contour plot consists of 3 clusters. We have 1 big cluster on the left and 2 smaller clusters on the right. And POTNet retains all 3 clusters. On the other hand, Wasserstein-Gann and OT fails to allocate enough weights to the smaller clusters, which is why you see the missing contours on the right-hand side. And this demonstrates the importance to both remove the discriminator network and also to include the marginal penalty as the regularization. In the second example, I will show that POTNet mitigates type 2 support shrinkage. So here, my data generating distribution is characterized by multiple modes and a truncated support. Again, I'm comparing POTNet against Wasserstein-Gann and OT. And if I put blue lines along the contours of the true data, we can see that POTNet faithfully retains the support of the data itself. However, you will see that there's a gap between the OT-generated data and then the blue lines, which demonstrates the support shrinkage caused by OT. So in 3 dimensions, this looks like the following. The original data has this round, voluminous structure around the 2 clusters, which is retained by PotNet. However, the synthetic data generated by OT lives in a thin hyperplane, and so there is significant support shrinkage along the phi-3 axis. We also applied PotNet to real data. So here, my data is the California housing data set, which comprises 10 features of mixed data types. I'm comparing PotNet against CT-GAN, which is a widely used alternative for tabular data generation. Here I'm plotting the bivariate density of house age and longitude. And I put red lines along the support of the real data. So we can see that PotNet generated synthetic data that accurately retains the structure of the original data because the first 2 plots look quite indiscernible to the human eye, but the third plot looks very different from the original data. So you can see that PotNet actually faithfully retains the structure. Finally, we can apply PotNet to digit generation. And we trained PotNet and Wasserstein-GAN on the MNIST digit data using convolutional neural network architecture. You can see that the resulting digits produced by PotNet is sharp and discernible, whereas Wasserstein-GAN produced several digits here that are not really discernible. So I've boxed those digits in red. PotNet is also very efficient in terms of data generation because of the architecture. So we simply push the noise vector through the generator network and we obtain our samples. So this makes PotNet about 80 times faster than CT-GAN and makes it very efficient for generating large amounts of data. Now in this last part of my talk, I want to briefly highlight the value of high-quality synthetic data. Or in other words, what can you use PotNet for? So PotNet can be applied to 3 main tasks. First is augmentation, and then evaluation and privatization. So for augmentation, we can supplement our training data with synthetic samples from underrepresented groups in order to mitigate class imbalance. When the event of interest happens too rarely, so that means we don't have enough training samples, we can also train the PotNet on these rare events in order to produce enough samples to train downstream models. We can also use PotNet to generate synthetic data sets that allow us to compare and evaluate different models and methods. And with high-quality synthetic data, we can systematically evaluate model performance without imposing stringent Monte Carlo assumptions when running the simulations. And the last is something we call privatization. This is actually a direction for future work. And the goal is to generate statistically similar but non-identifiable data sets so that we don't compromise individual sensitive information. And with that, I want to thank my wonderful collaborators and present to you the QR code if you're interested in using PotNet. Happy to take any questions now.